Good evening, and welcome you all for this IASTA seminar series. I'm Pandit Thurai, one of the executive members of IASTA. First of all, on behalf of IASTA, I am glad to welcome all the participants for joining this lecture by eminent scientist, Professor Rodney Weber from Georgia Institute of Technology. So first, I'd like to give some introduction about IASTA. IASTA is Indian Aerosol Science and Technology Association, which was established in 1988. The major objectives of IASTA is to promote and encourage and develop the growth of aerosol science and technology and their applications in the country, as well as to disseminate information concerning the developments in aerosol science and technology and their applications, further to organize workshops, symposia, seminars, courses, etc. in various parts of India to educate the users of aerosols and discussing new developments in aerosol science and technology, and also to promote international cooperation through exchange of information, joint workshops, meetings, symposia, etc. In aerosol science and technology. So now, with the able leadership of the current president of IASTA, Professor Sachidanand Tripathi, so we have several activities, outreach and training activities. And one of them is this monthly lecture series on aerosol science and technology. As you all know, the first lecture was delivered by Dr. Maya, a very distinguished scientist from Baba Atomic Research Center, Mumbai, and currently he is an emeritus professor at IIT Bombay. And this is a second such lecture in this series, wherein Professor Rodney Weber will be talking on wintertime air quality in Fairbanks, Alaska, outdoor and indoor air pollution under extreme cold in an urban environment. May I now invite Professor Sachidanand Tripathi, Professor IIT Kanpur and President of IASTA, to introduce today's speaker, Professor Rodney Weber. Over to Professor Tripathi.
Thank you, Dr. Pandit Thurai, and good evening, everyone. It gives me immense pleasure to introduce Professor Rodney Weber, who is an acclaimed international scientist. Professor Rodney Weber earned his BSc degree in mechanical engineering from University of Waterloo in 1987, MS in mechanical engineering from University of Minnesota in 1991, and a PhD in mechanical engineering from University of Minnesota in 1995. He served as an assistant scientist at Brookhaven National Laboratory during 1996 to 1998, and subsequently he joined the School of Earth and Atmospheric Sciences at Georgia Tech as a faculty in 1998. Rodney's research focuses on atmospheric aerosols and their impact on climate, air quality, human health, and the environment. He works on a wide variety of projects. Some of his current studies include particle emissions from wildfires, particles generated from roadways, and the unique chemistry of aerosols in the Arctic. He develops instrumentation and novel ways to characterize aerosol particles and deploy these instruments in airborne and ground-based studies. He has received numerous awards, such as Whitby Award of American Association for Aerosol Research in 2004, American Geophysical Union Ascent Award in 2014, and Bin Liu Award of American Association for Aerosol Research in 2016. He has published more than 300 publications, and he has an H index of 110. So with this, I will now invite Professor Rodney Weber to begin his talk.
Okay, thank you very much. You can hear me and see my slides okay? Okay. Well, thank you for the nice introduction. I really wish I was an eminent scientist. I don't think I really am, but it's a pleasure to give this talk anyway. I think this is an interesting topic because it's such a unique environment in Alaska in the winter. So I'm going to be talking about this study that was in 2022 in the winter. It was delayed because of COVID. So we're still in the process of analyzing this data. And I should say I'm showing results from quite a few investigators here, but I will talk mostly about the topics on this project that interest me. So see if I can advance my slide here. Why doesn't it go to the next slide? Okay, I have to use my other computer. So where is Fairbanks? It's in central Alaska. It's at a latitude of about 65 degrees north. That means it's very close to the Arctic Circle, where during the winter equinox, there's no sunlight for periods of time. So in the winter in Fairbanks, you can have some sunlight, but the sun doesn't get very high above the horizon for very long. It has a fairly small population, but it's kind of unique in that it's a fairly large population for such a northern city. And it's very cold in Alaska is the other thing. It's of interest because, as I said, it's such a high latitude urban environment. It's very cold. There's low solar radiation, and that leads to very few secondary particles or secondary oxidants, secondary processing. So the chemistry can be very different than other places. And this type of environment is actually not studied that much. And so that was really the point of this alpaca study, was to investigate air pollution in cold urban areas not studied because the chemistry can be significantly different. So the alpaca study had many interests. This schematic for the study overview kind of shows some of the emissions and the geography of the area. Fairbanks is in a valley. There are emissions from various sources. I'll talk about that. Some of the interesting chemistry they were interested in is sulfur oxidation, because sulfate is actually a fairly significant component of the aerosol. They're interested in gas particle partitioning because it's so cold and so very different. There was also a study, part of this study, that looked at outdoor air quality, because under these conditions, most people spend all their time indoors. So I will try to talk a little bit about most of these things, but depending how much time I have, I may not get to the indoor stuff. So one of the unique features of Fairbanks is that it has really strong temperature inversions during the winter. So I'm only going to talk about winter conditions in this study, obviously. It has very strong temperature inversions, and they're very close to the surface. So there's very little dispersion. And when you're in a valley, that makes things even worse. This graph shows a building in downtown Fairbanks at 20 meters height, PM2.5 concentration versus the 3-meter height concentration, as colored by the temperature difference between 11 meters and 3 meters. And you can see you get a difference in PM2.5 mass concentrations much higher at the surface, generally when the temperature inversions get stronger. And that makes sense. Strong temperature inversions, and you have little dispersion of the pollutants emitted from the surface. It separates actually the air that's above, that's more aged. Here's another way of showing that. This shows the PM2.5 concentration during the intensive study that I'm going to talk about. This is the time series from about January 13th, 14th to the end of February was the study in 2022. The PM2.5 concentrations in Fairbanks during some very cold events can get quite high. So this may not be high for India's or China's type air quality, but for United States, 60 to 80 micrograms per cubic meters over a period of time is quite high. Typical concentrations in Atlanta right now are about 10 micrograms per cubic meter. And this happens when the temperature gets particularly cold, but not always when the temperature is cold. There's sometimes when it's warmer. So this temperature here is like minus 30 degrees when we had the worst air quality. And if you look at the temperature difference between the 23 meters and 3 meters, you can see that there's this difference, which kind of indicates the strength of the inversion, is related to when we see these kind of poor air quality events. And you can get them actually during warm periods, but they're also important during cold periods too. The other thing, as I said, the oxidants are completely decoupled. So it tends to be that the ozone is higher above the inversion, and the PM 2.5 is higher below the inversion at the surface where people live. So this shows the ozone concentration at the surface, 3 meters, and the PM 2.5 concentrations. And this is the temperature inversion strength too. But you can see they're completely anti-correlated. And the reason this happens is because there's no photochemistry or very little photochemistry, which is very unusual. And the reason this happens is because the ozone that is around gets titrated from all the NO or the NOx that is emitted from the combustion sources at the surface that titrates the ozone. And there is no or very little sunlight to reconvert the NO2 back into, by photolysis back into ozone. So you get this very odd behavior of anti-correlation between the oxidants and the PM 2.5 concentrations. The other thing that's kind of strange about Fairbanks is the amount of wood burning there is. So people burn pretty poor quality wood that is not hardwood. They now a program where they make people burn dried wood, but wood combustion for domestic heating is a big source of domestic heating, along with burning fuel oil, which is basically like diesel fuel in a way. So although the chemical mass balance source apportionment studies are highly uncertain because the source profiles are not well known in Alaska at those cold conditions, for example, for vehicle emissions, wood burning always comes out as one of the major sources of the overall PM 2.5 in Fairbanks. We're trying to do better source apportionment in this study, but it's still pretty uncertain. Okay, so in this talk, I'm going to have 2 sections. I'm going to talk about this unique sulfur chemistry. I'm going to look at some preliminary observations of this compound, what we called hydroxymethane sulfonate, HMS, in the winters of 2020 and 2021. We did a pilot study and then COVID hit and everything got delayed. So the intensive study actually happened in 2022. But we have some interesting results from 2020. Then I'm going to talk about a more detailed analysis of the sulfur 4 chemistry. HMS is a component of sulfur 4. And then talk a little bit about modeling we're working on, particularly particle pH. And then if I have time, I'll talk about the air quality related issues with oxidative potential, indoor, outdoor. Okay, so why do we care about sulfur species in Fairbanks? Well, it's because it's about 20% of the overall PM 2.5 mass. It's tending to go slightly down because they're controlling the sulfur in the heating oil fuel and for domestic heating. The 2 main things they're trying to control in Fairbanks is the organic carbon or organic aerosol, which is a large component, which I said is mainly from wood burning, but also probably from some fuel oil and some vehicles, but there's not a lot of vehicles. The sulfur is mainly in the heating oil. There are coal-fired power plants, as you may have seen in this figure here, excuse me. There are like tall stacks from coal-fired power plants, but they tend to inject the pollutants above the inversion, and so they don't really have a large effect. We don't think they do. Ground level SO2 can be fairly high because of this burning of domestic heating. The sulfur, it tends to be largely unoxidized, at least when you look at SO2 to sulfate ratios, but there is still significant sulfate. EPA modeling, which doesn't do a very good job of predicting the sulfate, thinks that it's about half secondary. There's some isotopic work in this study that suggests it may be more like 80%, but it's not totally clear. But there is possibilities for unique sulfur chemistry in this location. So I'm going to show some preliminary results from the study. This is a picture I took when I was up there in 2020 setting up a PILS, a particle interliquid sampler. This is the entrance to the University of Alaska Fairbanks. This is an ice sculpture of a polar bear. This was at 5:30 in the afternoon. You can see that it's like, this is typically, it gets a little bit lighter during the day in January than what you see here. And the temperature was minus 33 degrees C. So it was very cold. So we had a particle into liquid sampler that collects particles directly into water, and the water is directly pumped to an ion chromatograph. So we can get an online measurement of the water-soluble ion concentrations. And one thing we saw, so this is from the preliminary study when I was up there just before COVID hit in March of 2020. So the people in Fairbanks there shut it down at that point, but then they ran it the next year again while we were still stuck in Atlanta. I took a picture with my phone of the screen of the computer connected to the IC, the chromatograph. And we saw this little peak here beside sulfate, coming out before sulfate. And we didn't know what it was. I had never seen it before. And at that time, there was some discussion about HMS in China, trying to explain this high sulfur and pollution events in China during the winter under high relative humidity conditions. So this compound, we bought a standard sodium HMS, and we put it in the IC, and we got a peak at this point. So we called it HMS, and that's what people in China have been calling the compound HMS too. They see it too. It's an interesting peak because you have to have the right IC eluent to see it, and we use a carbonate eluent, and so we're able to separate it from sulfate. If you use a different eluent, depending on the column that you use, most people do not have an eluent. If you use a Dionix IC, for example, I think it's a hydroxide eluent, this peak will not be separated, and so you will actually think you will measure HMS will be in the sulfate peak, and you can't separate it. It's also not quantifiable, at least so far, by an aerosol mass spectrometer, although you can see it with a single particle mass spectrometer, laser ablation instrument, but then it's hard to quantify. So it's kind of an elusive peak or elusive compound in more recent analysis of aerosols. So here is the data from these preliminary pilot studies we did. The gray here is the PM2.5 mass. This is in 2020 in January. The sulfate and the HMS are on a different scale than the mass, and this is the temperature on a scale up here. So you can see when it got pretty cold, it's pretty cold all the time during that period we were there, the masses can be quite high. Here it's like 60 micrograms per cubic meter at this one event. And the HMS, in what we call HMS, is in the blue, is almost the same as sulfate concentration, up to about 6 micrograms per cubic meter, which is high for our kind of standards here, our experience in the United States. This is January 2021 when Jingqiu Mao ran it for us while we were stuck in Atlanta. And you can see that again, we see HMS, or what we call HMS, and it tends to track sulfate somewhat, and it's high when there's a peak here. And so we can have fairly high concentrations of HMS relative to sulfate during these pollution events, and it can be a significant fraction of the mass of PM2.5. So HMS is kind of interesting because you go into a textbook like Seinfeld and Pandas, it's all discussed there. In the 1980s, it was the chemistry was looked into quite significantly because of interest in cloud chemistry. And so Hoffman, for example, has written many papers on HMS in clouds. It's been seen in other places when they do detect it, but the concentrations are generally low. And this can be because if you have oxidants around like OH, it will convert HMS fairly rapidly, depends on the OH concentrations, into sulfate. So this could be a route in which some sulfate is formed through this chemistry instead of the typical hydrogen peroxide ozone chemistry that makes sulfate heterogeneously. In China is where they've been looking a lot at haze, winter haze when there's high RH, and they can get very high HMS concentrations in the winter. And it can be a few percentage of PM2.5 mass. And the actual HMS concentrations are higher than what we see, but then their PM2.5 concentrations are in the hundreds of micrograms. So we actually see a much higher mass fraction in Fairbanks than they do in China. So why is there so much HMS in Fairbanks? The chemistry of HMS is essentially that it reacts with formaldehyde. So this is an aqueous phase chemistry where the SO2 goes into the droplet, whether it's a cloud droplet and or maybe a particle, liquid water. It's hydrated, it breaks up into bisulfite and sulfite, and the formaldehyde also partitions into the water, and they can react. Here, the formaldehyde can react with either the bisulfate or the sulfite and make HMS. There's a strong pH dependence on this because these concentrations of the bisulfate here and the sulfite here depend on pH. So it's a lot like the chemistry of ozone oxidation of SO2 versus hydrogen peroxide oxidation of SO2. But in Fairbanks, there's no ozone and so no hydrogen peroxide. Another interesting feature is that you can have these liquid droplets possibly down at really super cool temperatures. So HMS is actually a tracer for liquid phase chemistry, which is kind of interesting. But we found out when we looked at this more detail, we started getting ready for the intensive study in 2022, that sulfur species in general coelute in the IC that we're using at the same time, or coelutes at the same with HMS. So if you make a standard of a bisulfite and HMS, you get the same elution, peak at the same time. It elutes at the same time. So you really can't separate these two. So what I had been calling HMS and what people call HMS in a lot of these other papers is probably HMS plus maybe something else. We should really just call it sulfur 4. So that's what I will call it now. But there is a way to separate HMS from what we think most of the other sulfur 4 species like sulfite and bisulfite adducts. So if you add hydrogen peroxide, and we only did this to the filter samples, if you add hydrogen peroxide to your extract, it will react with so you extract a filter and water, and before you do the IC analysis, well, you do an IC analysis with just straight sample after it's filtered, or then you take another part of the sample of the extract and you put in hydrogen peroxide and you let it react for a little bit of time. We use a flow system to do this, so we do it automatically. But you can do it just in a beaker. And that hydrogen peroxide will not affect the HMS concentrations, but it does seem to affect many other bisulfite sulfur 4 species that get converted to sulfate. So that's the way you can try to separate out HMS. So for example, we run offline on the filter, a sample total, we get the HMS here. It's not separated the best from sulfate peak. Then we add hydrogen peroxide, the peak drops here, and that peak area is now related to the HMS. And what's the difference between these two is what we would call other sulfur 4 species. So now focusing on the measurements in Fairbanks, the instruments, we had a whole suite of instruments there. There were many investigators from Europe and North America. I was in this trailer. We had a pills here that was right up against the wall, insulated inlet to keep everything as cold as possible. So there's no temperature change until the aerosol got right into the instrument. The filter samplers were located outside so that we didn't lose samples. There's major challenges in trying to collect particles outside and inside. And when it's this cold, you can't use, for example, most plastic tubing, it just shatters. So there's many sampling issues here. But anyway, we measured with the pills online sulfur 4 because we didn't do the peroxide separation, the various ions, and we did, it has a 30-minute time resolution, etc. There are measurements of formaldehyde, SO2, meteorological parameters, many other things, AMS, ACSM, things like that, single particle mass spec, many measurements. We also had filter collections. So we had a PM 2.5 mil filter collected every day outside. University of Washington, we're doing Becky Alexander, who's doing isotopic analysis of sulfur to figure out if it's primary or secondary. Had a high vol sampler that had some size segregation. I won't talk too much about this, but I'll show some results from her lowest size, 0.7 micron. The University of New Hampshire measured total suspended particulate particles on a filter at time resolution less than 24 hours. Sometimes it was 4 hours. And we also had a Moody sampler, but I won't show that results. Those results are kind of interesting, but I don't have time to show it. So this is equivalent kind of what I showed you before for the intensive study, which was in January 17th to roughly the end of February. Here's the temperature in green. It was, typically below 0 degrees Celsius. Sometimes it got down to minus 30 degrees. During the really cold events, we had very high sulfate for our levels here in the United States, 14, 15 micrograms per cubic meters at that time. The HMS, I should call it not HMS, the sulfur 4 measured by the pills is quite high. I should say that this is the only measurement, I think, online measurement of sulfur 4 that's quantitative with a liquid-based system like this. So this data is kind of unique. During this event, it was really cold. That's why we have very high concentrations, strong inversions. People are burning a lot of wood to stay warm. The sulfate is around 16 micrograms, and the sulfur 4 was around 14 at most. So what about this sulfur 4 and sulfate and low temperatures? There is an enhancement. So if you plot sulfur 4 versus sulfate, color it by temperature, you can see that they both go up when together. But there's a nonlinear relationship here between sulfur 4 and sulfate. If you make a graph of the ratio of sulfate or sulfur 4 to sulfate versus temperature, you see that as the temperature gets lower, the fraction of the sulfur 4 to sulfate actually increases. So there's the online data in red, and the filter data is the big gray dots. There's more scatter, of course, in the online data because it's not averaged as much. But there is this strange or interesting increase in sulfur 4 relative sulfate with lower temperatures. And this generally happens at the most polluted periods when the temperature is the lowest and the pollution levels are the highest. We can look at HMS to sulfate. So when we did, this is just for the filters, where we did the hydrogen peroxide treatment to separate HMS out from the other sulfur 4 species. And you see this trend pretty much too. So what is the effect of temperature on what's going on here? So there's an increase in sulfate in HMS. And in fact, everything that's emitted at the surface increases when the temperature gets lower because people are burning more to stay warm. There are strong temperature inversions. There's high pollution levels. But why does sulfur 4 increase relative to sulfate? Well, there may be more sulfate around, so there could be more liquid water, possibly even down at minus 35 degrees C. So this chemistry is going in very cold, super cool droplets of water. And I should say the unique thing about Fairbanks relative to other places like China and Beijing, for example, in pollution events is it's likely that most of that chemistry is happening in clouds and fogs. Fairbanks is unique in that it is happening, we think, in particle water. The thing that's driving this is probably that the solubility, the Henry's law constant, or more really better to think of it as the volatility of the precursor gases that react in these liquid droplets, their solubility, or they want to be in the liquid phase. And the Henry's law constants can increase by significant factors when the temperature gets this low. There's also an effect of temperature on particle pH, because you need a pH, as I'll show you in a bit, of 4 to 5 to get this to happen. So what about HMS to sulfur 4 fraction? That's pretty linear. And it's depending on what filters you use, what size ranges, there is somewhat of a bit of difference in this ratio. So this is a mass ratio, not a molar ratio. So this slopes for these different size particles captured on filters, it's all about 50% to 65% of what the sulfur 4 we see is actually HMS. So I shouldn't really have called it HMS in the preliminary data, but it is correlated with it. And this has actually been seen in cloud droplets. Jeff Collette, back in the day when people were more interested in HMS in terms of cloud chemistry, they also see that there is, they only measure a fraction of the HMS is only some fraction of the sulfur 4. So what is this non-HMS sulfur species? Well, there were papers on this topic some time ago. Ita published a paper in 1978 where he looked at the sulfur 6 pool and the sulfur 4 pool in aerosols. This is when people were really into power plant emissions. And one thing in the sulfur 4 pool, which HMS is a part of, where sulfate is part of the sulfur 6 pool, is that there could be metal complexes. And this was more like in power plant plumes associated with fly ash and things like that. But we measured metals in Fairbanks, and they're actually very low concentrations relative to many other places because vehicle emissions are so small relative to the overall emissions in Fairbanks. If you take sulfur 4 minus HMS versus water soluble iron, for example, the molar ratios suggest there's just way too little iron to be, and this is the most abundant of all the metals. There is very little iron, and not enough iron to account for all of the missing sulfur 4 or unidentified sulfur 4. It could be other adducts. So we focus on HMS, formaldehyde reacting with the sulfite or bisulfate to make HMS. You could have another aldehyde that makes does the same thing and makes an adduct too. And so we're looking into that now. And this has actually also been studied by Hoffman et al. Back in the day in the 1980s. There are other aldehydes that are fairly soluble too in water, so they could be taken up just like formaldehyde. They're also probably emitted in Fairbanks, but not at highest concentrations as formaldehyde. And the most prevalent one is acid aldehyde. So you can have acid aldehyde react with, for example, sulfite and make HES, hydroxyethane sulfonate. And the thing about this is when this is less stable in HMS, so when you put it in water, either a pills or but we did this for filters, in the filter extraction, HMS will not fall apart. It will stay as HMS, where this HES or other adducts will fall apart, and then we measure the sulfur 4 species, and we don't measure the aldehyde species. So this could explain why we are missing what the missing aldehydes or the missing sulfate, missing sulfur 4 is. And it could actually kind of explain the linear relationship we observed, where the HMS to adduct ratio would be related to the gas phase concentrations in this kind of simple idea. So we detect the HMS and the sulfite or bisulfite left over after that adduct has fallen apart. Why doesn't it switch here? I can't advance my slide for some reason. Okay, so the single particle mass specs can actually see these species. So the HMS here is one mass to charge or molecular weight of 111. And this is a mass spec data I got from a paper in Beijing. And they didn't really talk about it very much, but they have a mass at 125 that actually is the molecular weight of the ion of HES. It's hard to say what the concentrations are from these data because we don't know how to convert the relative peak area to a mass concentration. So these other aldehyde adducts probably exist, but I don't think we're working on this problem with Jason Surratt trying to get better quantitation of these aldehydes. It's very hard to do actually, so I don't know if it's going to work out. But it could be that they are a significant contributor, but I think they're a small fraction and won't explain the most of it. So it still is kind of a mystery what these other species are in the sulfur 4 that are not HMS. They could be And that, it could just be an artifact that the HMS falls apart in our IC and goes to sulfate, and or just goes to it falls apart in our IC, and so we don't really see the HMS. Okay, so I want to talk about modeling very quickly here. So we're working with JinQ to model this chemistry, and it turns out that the route for the HMS to be formed is through the sulfite reaction, because the rate constant is much higher than with the bisulfite. And you can also possibly make secondary sulfate through this same type of reaction. But let's focus on this chemistry. This makes a very strong pH dependence on the chemistry for it to occur, because the sulfite concentration depends on pH. You can't have a pH too high or the adduct falls apart. It's the sweet spot is somewhere between 4.5 and 5.5 of pH of what you need to make this adduct. So what is the pH in Fairbanks? So we ran isopropia. We used our pills ion data and some filter data too. We had a mist chamber to measure the total ammonium. We used that instead of trying to measure gas particle ammonium, because we were worried about volatility issues in the measurement when we brought this sample indoors. If you run isopropia, you get 2 particle pH kind of ranges. You get this kind of bifurcation. It's either very acidic or the pH is more neutral, 3 to 5. This is the range in which you can make HMS. In this range, it will be stable, but you will not, because there's no oxidants, but you can't really make HMS in that range because the sulfite concentrations are too low. If you look at a time series of pH, it jumps all over the place. It goes either between very acidic to somewhat neutral. So this top pH range is when you can make HMS, but down here you can't make HMS. So in all the studies I've done of particle pH, I've never really seen this, unless you're in an airplane going in and out of a plume or something. I've never seen this kind of bifurcated jump. And this is pretty interesting, and it's unique to Fairbanks, and it's actually unique to cold temperatures. So this is a sensitivity test of particle pH predicted by the thermodynamic model, isopropia, showing the sensitivity to temperature. So these are each different temperatures. And it's the ammonium, it's total ammonium to total sulfate in the model for different temperatures and kind of for a given liquid water concentration that we calculated in Fairbanks. Other species like nitrate are so low, we don't really have to consider them as non-volatile cations like calcium and things like that. That does not have a big effect on this. What you see that's very strange here is that as you get colder, you can see there is this jump. And if you pick a certain sulfate concentration, say 10, and you go up in ammonium concentration, total ammonium, gas plus particle, you reach a point. So if you do it at a warm temperature that I'm more familiar with, the pH never really gets very high. It's always around 1, 2 or so. That's typical Atlanta type pHs. If you get to colder regions, the pH is still low at certain ammonium to sulfate ratios, but it suddenly will jump to a higher pH. So you can go from like a 1 to 0 pH with a small increase in ammonium or a total ammonium, you can go to a pH of 4.5 or 4, 5. And this happens with temperature. And the reason why this happens is because sulfate is not volatile, but ammonium is. And so as it gets really cold, the ammonium partitioning is driven more towards the particle phase. And so you get more and more ammonium partitioning, and the sensitivity changes to the ammonium to sulfate molar ratio. And in fact, it's known now, I mean, we wrote a paper on this, that you should not use ammonium to sulfate molar ratios to predict pH, because on this graph, if you would convert this to moles, the molar ratio of 2 is somewhere aligned like around here. And you can have a molar ratio of 2 and be very acidic under warm conditions, depending on the other parameters involved. But when you start to get to really cold conditions, you get to that actual situation where molar ratios actually start to predict the pH, whether it's acid or base, pretty closely, because If you jump below the molar ratio of 2, just slightly, you become very acidic, or above, you become quite neutral. And this is a unique feature of cold temperatures. It's somewhat analogous to adding huge amounts of liquid water, going from an aerosol phase water to cloud water, where then all the ammonia wants to be in the condensed phase. But here it's driven by volatility in the cold temperatures. And so that probably explains why we have this jumping in pH and why we get these conditions every now and then conducive to make HMS. We can see HMS at other times down here because it's stable. And so these are probably conditions under which HMS was not being produced. Okay, so the end of this section is HMS is a significant component of the sulfur species at low temperatures on these unique conditions of no oxidants. Sulfur 4 is formation relative sulfate is enhanced at low temperatures, probably mainly due to, since HMS is only produced through this heterogeneous chemistry, it drives the gases into the particle, increasing this heterogeneous chemical reactions. HMS is only about 1/2 of the sulfur 4. It could be the other part of it is other aldehyde sulfite adducts. We don't really know. I don't think it's going to account for a large fraction of that, but it's an interesting problem. It could be an artifact in the IC method of measuring HMS. Particle pH behaves very differently in cold conditions due to the sensitivity of the ammonium partitioning with temperature, and that can create the conditions conducive to making HMS. So we're continuing to work on this. This chemistry is pretty interesting because it's so unique in Fairbanks, because we really believe it's happening in PM2.5 particles, not in clouds, which is where everybody else has looked at HMS in the past. It also may be that in other locations, this is important. So it would be interesting to go to other cold cities and see what you see for this kind of chemistry. It may be where you have oxidants around cold cities that have more sunlight. It could still be important because this could be a route. Although the HMS concentrations may not be as high because they're oxidized eventually, it could be a route for oxidizing SO2 to sulfate too. So in the last, I think I have about 10 minutes, I will go on to the outdoor-indoor air quality and oxidative potential. So not far from the downtown area, there is a little residential area, and we rented an Airbnb. And we had some measurements outside this house, and we had a filter sampler inside the house. So I'm going to talk about oxidative potential both outside, and if I have time, I'll talk about inside. So why do people care about oxidative potential as a metric for the possibility of an aerosol leading to an adverse health outcome? Well, the problem is with PM2.5 mass is it doesn't consider the variable toxicity of the specific species in the PM2.5. And oxidative potential is supposed to mimic the process at which particle species deplete antioxidants and make oxidants within cells in vivo. And those oxidants cause an oxidative stress response, which can lead to many different types of adverse effects: cardiovascular, respiratory, dementia, things like that. And there are specific species that measure that can cause this oxidative stress response. And it's thought to be things like some SOA species can directly produce OH, but the more catalytic production of oxidants is through transition metals and quinones, things involved in redox chemistry, which can make superoxide that converts to hydrogen peroxide, and then through maybe more Fenton chemistry converts hydrogen peroxide to OH, and then that causes problems. So it's more biologically relevant, possibly, than PM2.5 mass, but there are limitations because different assays are sensitive. Assay being the chemical method you use to try to integrate these various compounds into one measurement. And they tend to integrate or are sensitive to specific species. So I'm going to talk about the DTT depletion assay, dithylfrito depletion assay we use, and an assay that measures OH production in synthetic lung fluid. There were other measurements there, for example, of environmentally persistent free radicals was done by Manabu, but it turns out that that is very highly correlated with the OH production assay. They did also ROS generation, but I won't talk about that today. So at the house, here's the composition of the aerosol, as I showed before. We're now going to focus on these very minor species of metals that are only about 1 to 2% of the aerosol. The AMS was there, Pete DiCarlo's instrument, and they speciated the aerosol, the organic aerosol, into biomass burning organic aerosol, hydrocarbon organic aerosol, and this other factor that they just called primary organic aerosol 2. So the interest here was on these, the research questions are, what are the major chemical species driving the aerosol toxicity? How does Fairbanks and OP compare to other locations? And do these measurements give a different view of particle toxicity than PM2.5 mass? For the indoor study, we wanted to see what happened when air infiltrated the house, because there's a large temperature change from -20 outside roughly to say 20 degrees indoors. And everybody likes to do these different indoor cooking experiments or something and see how things change. So I'll talk a little bit about that if I have time. So to address the question of species, we did a multiple linear regression. And what we find is we did 2 regressions. We could have done a non-linear regression, but we get our good R-squareds with just a simple linear regressions. And we've done that with various species, and we get generally that we get the same kind of sources involved, same type of emissions that produce similar species. So in unstandardized regression, we have the coefficients represent change in OP per unit increase of the independent variable. So in this case, the total DTT, that's all the species we think, including insoluble species, copper and PAHs, which is a factor from the AMS, are the main components that contributed to total DTT. But the loading, the coefficient here in front of this independent variable, copper, is much higher than the PAH coefficient. For water-soluble DTT, the copper, it was copper and BBOA. So water-soluble is a fraction of the total DTT. The OH generation assay was the hydrocarbon organic aerosol was important, and water-soluble iron was important, as was total iron. So this result shows that these assays are really most sensitive to transition metals compared to organic species. But the OH, and this is particularly true for the OH assay, where the DTT assay is more evenly, these coefficients are probably closer together, you would say. This is well known, but it's true when we found in Fairbanks too. Now, if you do the standardized regression, this is actually considering the concentrations of the species. So this is more of what's important from an exposure point of view. Then the organic components are more important. The coefficients are bigger because there's just a lot more PAHs than there is copper or BBOA organic aerosol components. That's biomass burning organic components. And the OPV is also that's true too. So what we find is kind of what's been known before, but in Fairbanks, we have these specific species. There's a contrast in what these assays measure. I won't show the correlations or other data to suggest where these species come from. I'll just tell you the PAHs are mainly relating to biomass burning emissions and some from the vehicles, mainly diesel, we think, based on correlations with other species. So it's mainly from probably wood burning. The HOA and the iron is coming from vehicles mainly, and the copper comes from many different sources. Now, this question of is Fairbanks uniquely toxic aerosol? So recently there was a paper by Suzanne Paulson talking about LA. And so we took her data and we added it to our data from Atlanta. And we looked at it in comparison to Alaska. This shows PM2.5 mass averaged for a winter season in Fairbanks, Atlanta, and Los Angeles from Suzanne Paulson. So this is just winter data from her paper. You can see there's lots of variability in the PM2.5 mass, which I talked about due to temperature inversions and things like that, meteorology. But from a mass point of view, Fairbanks is more polluted. From an oxidative potential based on a volume normalization, so its exposure on the DTT assay, Los Angeles is actually more polluted or more hazardous. And on a per mass basis, which is kind of like the intrinsic toxicity of the aerosol, it tracks basically the volume too. So the aerosol is actually from this assay suggesting it's more toxic. You would not get that impression based on PM2.5 mass alone. But the DTT assay is more similar between Atlanta and Fairbanks. Whereas if you look at the OP-OH generation assay, shows that LA is much worse than all of them. And so this is simply because these two assays are sensitive to different species. The DTT assay tends to be more sensitive to organic species, so the high biomass burning contributions in Fairbanks contribute to this high assay. But this assay, the OH, is less sensitive to it. It's more sensitive to vehicle emissions, the HOA in Fairbanks and the iron. And so because there's so few cars in Fairbanks relative to these other cities, it gives a completely different view of how toxic the aerosol is and what the exposures are. Okay, so one last thing about the indoor-outdoor air. They had a pellet stove in the house that they installed. This was an Airbnb, it was empty. The instruments were separated from the house in the garage, and they put a fake door in and they sampled through the door. They also did some cooking experiments. I will skip this slide. I just want to show the results here. If you have nobody in the house, there is infiltration of the polluted outdoor air into the house. And if you use sulfate as a measure of an inert species to get some kind of idea of what the rate of or the level of this infiltration is, about 20% of the outdoor air seems to infiltrate the house and contribute to PM2.5. So the ratio of outdoor to indoor sulfate is about 0.2. The DTT measured with a filter, it's about the same. So we're only getting the non-volatile DTT species, such as quinones, metals, transition metals. It behaves very similar to the sulfate, which is not surprising. If you take the ratio of the mass normalized, that is, you mass normalize the outdoor and the indoor, and you take that ratio, it's about 1, which means the toxicity of the aerosol didn't really change based on this assay measured with a filter. If you run a pellet stove, you get significantly higher toxicity of the aerosol relative to outside. If you burn incense, you get somewhat higher toxicity relative to outside. If you do cooking that you don't burn your food, you get actually very innocuous aerosol. It's not that toxic by this assay. So my point here is that the question of whether PM2.5 mass is a good metric, actually, when you do indoor experiments, it's probably even a worse metric than when you are looking outside aerosols, because outside there's many processes that can make correlations between species. And inside, if you would just looked at mass, you would have said that the cooking experiments produced about as much mass as when we're running the pellet stove, but there's a completely different response from this assay. And I think the other assays that were indoors get the same thing. So my last slide shows OP assays are sensitive to transition metals. We've all known this. Fairbanks OP is similar to Atlanta. So in terms of toxicity and the exposures, in terms of OP, it's actually not much different than Atlanta. And LA is actually worse than them all. OP does seem to give a different view of air quality than PM2.5 mass when you look at these broad comparisons, highly averaged data. The indoor data that really, I think the take-home message here is that, of course, as one would expect, what actually that activity is indoors is really important in terms of the toxicity based on these OP assays. Okay, so this was funded by NSF mainly, a bit by NASA. I worked with Jing Q and his graduate student James, and my postdoc Kayane is helping me with this project too. This is a nice picture I took from one of the sites. You can see how that you get very different behavior in where emissions are occur at and what altitude. Okay, so that's it. Thanks. I hope I didn't take too much time.
Oh, you have taken not at all enough time. So you were finished right in time. And thank you so much for such an interesting and informative talk. I'm sure there'll be many questions. So I request the audience to raise their hand and also request the moderator to allow people who are raising hand to ask question directly. So people can raise their hand. Okay, there is a question in chat. It is by Jay. He is a student. So he's asking, hello, sir. I want to know that HMS that you got in IC, is it because of the so-called ambient temperature of the site, or it is because the precursor availability is more for its formation? So it means he's asking whether it is because of precursor or ambient condition. So which one is more? Yeah.
Precursor or ambient conditions. It's ambient conditions. It's the cold temperature, because you can see that in the data set. The concentrations of SO2 and formaldehyde don't change a huge amount during the study, and the temperature generally got warmer as the study went on. But the HMS was more prevalent during the colder periods. So what it really is, this is why it's so interesting, I think, is that this chemistry will normally not occur to much extent in a liquid droplet, because there's so little water to absorb that the gases can diffuse into and for the chemistry to occur in. But when you get it really cold, then the volatility of these gases, that is SO2 and formaldehyde, drops so much that it increases their Henry's law constants tremendously, like factors of, I don't know, 6, 5, maybe even 10, I can't remember the numbers, which drives the gases more into the particle phase, and that's what makes the chemistry happen. And so the model is that it's kind of rate limited by the uptake of the gases. And that is driven by the low temperatures. So like I said, I would probably bet in India, in colder seasons, when you have very high relative humidities, even though it tends to be warm and you have photooxidants around, or like OH radicals around, you still might get HMS like they'd see in Beijing, because it's happening in big, large droplets, possibly fogs or clouds. The unique thing about Fairbanks is it's happening in very little liquid water. And it's because the temperature drives the gases into the liquid phase.
So do you think, you said that over the Fairbanks there was no hydroxyl radical, but if we are in the site like India or this kind of site, you will have too much of oxidants also. So will they allow to this HMS remain there, or it will oxidize it to sulfate?
Well, I think it will eventually oxidize it to sulfate. So you may actually have a lower concentration relative to the mass, but it's kind of like, it takes time for the OH to oxidize it. So I think that's why they see it. And there's papers on this. It's been modeled, and there's papers in ACP on this and stuff in China. So they conclude that it's all in the cloud droplets. So it's you will see it. You will see HMS, or what we really should call sulfur 4, because they don't separate it from the sulfur 4 from the HMS from the sulfur 4. You will see it, but you're making it faster than you're losing it probably for a period of time, and that's why you see it. But that doesn't mean it's not an important route for converting SO2 to sulfate, right?
Right.
But in Fairbanks, we don't have oxidants, and that's why we can get such high concentrations of it too, relative to other places.
Yeah. Okay. Anyone else? If you raise hand, moderator can unmute you, and then you can ask the question. Okay, there is another question by a student, Chandrima. She is asking, did you run isoropia in reverse mode? If so, did you calculate the pH from the H plus concentration or consider the pH value given by isoropia?
Yeah, so the only way we ran isoropia and we calculate the liquid water. We've done it with including the organics to determine the liquid water. It doesn't make a huge difference on the pH. And so that the pH all the pHs I've shown, which of course is always the case. I don't know of anybody that can accurately measure pH in these size particles, is all in model prediction. So then what normally you can think of this as actually two different ways. If the chemistry is right and you have to have a pH of between roughly 4.5 and 5.5, then when you see the problem is that HMS can stick around. So the conditions you measure it under doesn't necessarily mean that's the conditions it formed under. But it does suggest that there are places in Fairbanks where the pH is in that kind of a range and there is liquid water present because you see HMS. You see it in the particles and it goes away and it comes back and it goes away. So I guess my answer to the question, if I understand it, is yes, everything is predicted with isoropia. It would have been nice if we would have had better, more specific gas phase ammonia measurements so that we could have actually checked the model predictions by looking at the predicted partitioning of gas particle ammonium relative to what we would observe. That's what we normally do. We could have done the same with nitric acid nitrate, but those measurements weren't there. And they're extremely hard to make under these colder conditions when you're sampling outdoor air into a sampling building.
Yeah. Okay. Thank you. So anyone else? Okay. Means until somebody is thinking, let me ask one question. So it is related to the oxidative potential results you were talking about. So people talk about this synergistic and antagonistic effects. So at present means we are looking, say for example, you were showing this multi-regression analysis, and some species you are able to see that copper or PHS or HOA. But people also talk about there is a synergistic effect. So that means those two species individually they are not so toxic, but when they are coming together, or same way antagonistic effect. Individually they are toxic, but when they are both are there or more than two are there, then they are neutralizing the effect. So what are your views on that?
Well, yeah, that's Vishal's has done a lot on this, and
Yeah,
we've written a couple of papers on this in Atlanta too, and I'm sure there are effects, both antagonistic and synergistic effects between two species. We didn't do it because I guess the graduate student, I was just talking to her yesterday about this, we'll look at it, but I don't think it will change the conclusions too much. We did get, our R-squareds are 0.85. So we do a pretty good job of predicting the variability with this very simple linear model. Yeah. And there's something to be said about a simple linear model. So it's easier to explain. The big thing that worried me more than that, but I agree with what you say, Niraj, we should do that in more detail. But the bigger thing is because there's a lot of covariability in these species, because everything is kind of controlled by the inversion height, that some of the things we have to select which ones to include in the regression, which species. So we've we've done different combinations in the linear regression, and they all point to similar things. Either the biomass burning and the copper, the metals pretty much stay the same. Sometimes the PAHs can be replaced by like black carbon plus a biomass burning marker, like brown carbon. I didn't say anything. We measured brown carbon here, and you can use brown carbon instead of the AMS data, and you get very similar results. For example, BBOA is really highly correlated with brown carbon, as is the PAHs are pretty highly correlated with brown carbon, where HOA is really highly correlated with CO, but not so as high correlated with brown carbon. So you can use different variables and you'll get kind of similar results. But I would say in terms of the synergistic and antagonistic effects, we should probably run the more advanced model and see what it does. But at this point, I kind of just like the simple result.
Yeah, thank you. So anyone else? Ms. Moderator, Ms. Can you allow to unmute people if they want? Now it is the interaction time, so they can, I think they are not able to unmute themselves. Okay, can you please allow them? Okay. All are unmuted. So people are unmuted now, so whomsoever want, he or she can interact directly. So you can unmute yourself, as that is what moderator says. Anyone? Professor Sareen, would you like to interact? No, Miss, why I'm telling him because I want to introduce him also if he is there.
No, thanks. I think it was a very interesting topic. I think I would rather be more interested in the looking at the oxidation potential which he was talking about, and the pellet stove was looking very interesting. So I don't have any specific question on that, but it's very surprising to see that the ratio is very high between the indoor and the outdoor. So I think that's something which is very important to see that. Thank you.
Okay, thank you.
Another way to look at this, I skipped this graph for time, is you want to talk about the pellet stove. This graph shows DTT normalized by mass. And this is when there's no experiment for the sulfate, and this is the DTT. But this is what's kind of interesting for the indoor experiments here, is that this is the pellet stove, the DTT per mass. So you can take the DTT volume and you divide it by the mass of the aerosol in the room. When the pellet stove was operating and it leaked a little bit, you could smell the wood smoke if you walked into the room. They didn't install it correctly for us. And then this is the cooking oil. So it went from like 0.01 nanomole per minute per microgram to up to about 0.9. So it's like a factor of 9 increase when we went from the pellet stove to cooking. I mean, it really emphasizes that it's the combustion, incomplete combustion components from in the organic fraction that drives a lot of this toxicity. So this oil drop experiment was, they just put, they either put water into a hot pan of olive oil, or they put like a piece of chicken or something, and it made splattered a lot of oil around and made the little fine particles of oil. And of course, you would not really expect olive oil to be toxic. And so in the home chem experiment, people have talked about toxicity of cooking based on mass, and they kind of equate it to mass. But I think that's really, in an indoor environment, that's really, really not probably the best way to do it. In the outdoor environment, it may be a better correlation between mass and toxicity, actually. But an indoor environment with a specific activity where you have a very, very strong source and not much else, I don't think that really applies, because it is really essentially the metals and the incomplete combustion organic components that drive much of these assays. And that's pretty consistent with what people knew in the past were the toxic components of aerosols.
Okay, thank you. So another one is there is a person, Ashish Soni. He is asking what could be the chemistry for such an acidic pH in Alaska. So since you are going in less than 0, also probably he want to understand that why pH is going so low.
Oh, why it's so acidic?
Yeah.
I think it's just because there's not a lot of ammonium, I guess, is, you know, and the amount of liquid water you have. I think that's,
yeah.
Yes. And another way to look at this is through an idea of buffering. Buffering being that, you know, a buffering by ammonia partitioning between the gas and the particle phase. And when you get to, or you can think of the buffering of the sulfate going to bisulfate back and forth, because that changes the H plus concentration going from HSO4 to SO4. So the buffering kind of switches to the sulfate bisulfate when you're at the low pH, and there's not a lot of, that's at that point, all the gas phase ammonia, when you have such low pH, the ammonia is all taken up by the particle. And so you don't have any buffering by ammonium partitioning.
It is lack of neutralizer, I think, mostly ammonia or any other neutralizing components are not there. So that is why it is acidic. Yeah.
Yeah, so it's, but when you get to higher pH, then the buffering is by ammonia. But as you get closer, lower and lower temperatures, that buffering kind of changes, because eventually it goes to where there just is no gas phase ammonia anymore, because it's so cold that it will all be in the particle phase. And then the pH goes high, and we don't actually ever get there. And then I think the really interesting thing about that plot, those plots I showed you, is that the idea of a molar ratio, ammonium to sulfate, it clearly shows it does not tell you anything about pH. It does not tell you anything about pH in warm environments. But when you get to extreme cold environments, it actually does start to, ion balance does actually start to tell you something about aerosol acidity when it's really, really cold.
Yeah.
And that's unusual, because, you know, people have been often using ion balances, I did too, and to talk about pH, but it really is not an accurate, it doesn't tell you if it's acid or not. It doesn't tell you. You can have a molar ratio of ammonium sulfate close to 2 and the pH is 1, you know, depending on the conditions, the temperature and the relative humidity. So that's what makes Alaska, the pH in Alaska, so interesting. I think it's like I said, it's almost analogous to thinking about what happens when a particle becomes a cloud droplet. Then there's no gas phase ammonia either, right? Because there's so much water to absorb the gas ammonia.
Right.
And when you get really cold, what drives it is volatility, not the unlimited amount of water to absorb the gas. Now it's so cold that the volatility just drives the ammonia into the particle phase.
Yeah, thank you. So is there any other question? Okay, it seems not. So it is the time of now vote of thanks. So let me take this honor of telling the vote of thanks. So firstly, on behalf of IASTA and on my own behalf, I wish to thank Professor Rodney Weber for kindly accepting our invitation to deliver this lecture. While contacting him for the first time, actually, I made a mistake. Means I wrote that it will be 9:30 a.m. for him. And he agreed. And then afterward, I realized that it is not 9:30, but 7:30. So for him, it is actually 7:30 a.m. So he got up so early and he is giving time to us. So thank you very much for that. And when I said it was my mistake, he simply said, no problem, don't worry. So he kindly agreed. Thank you so much for the giving time to us, as well as enlightening us with your wonderful talk. I also wish to thank the team of BARC led by Dr. Manish Joshi, who did the great job of handling Zoom. Actually, we just started this online lecture, and this is the real first online lecture. Earlier one was the hybrid mode. So we are still, kind of learning on this, and they just got 2 days before they got the Zoom, and then they learned and they handle this. So their efforts are appreciated. Lastly, I wish to thank the whole IASTA team led by Professor Tripathi for their continuous support. And you might have noticed at the end of the slide of Professor Weber, means that was the announcement of our next upcoming IASTA conference, which will be in Mumbai during 12 to 14 December 2023. So please block the dates and please join us with your latest results. And with this, I wish to thank you all. Thank you very much for your time.
Thank you. Nice seeing you, Neeraj.
Yeah, really, it was really, really nice to see you after such a long time.
Thank you, Professor Weber. Thank you. Thanks a lot.
Okay, so we can end the meeting now. So we can end the meeting now.